One of the major factors to maintaining homeostasis within the body is hemostasis, which is the body's response to injury. The process of hemostasis has three major phases, starting with vasoconstriction. Vasoconstriction begins with the baroreceptors signaling the brain about a drop in blood pressure. In response, norepinephrine is released into the tunica media surrounding the blood vessels. Within the smooth muscle tissue, norepinephrine is transducted, resulting in activation of the signaling molecule IP3, which goes on to moderate the release of calcium ions from the cell's sclerosing reticulum. Additional calcium ions are taken into the cell via active transport channels. The newly released ions combine with caldonin, forming a complex which then deactivates calponin and caldesmin, allowing the myosin and actin to interact. The complex goes on to form a myosin light chain kinase, which places a phosphate ion on the tail end of the myosin head. ATP bonds to the myosin head, allowing it to detach from the actin. The phosphorylation of the myosin head allows for the decomposition of the ATP into ADP and inorganic phosphate, with the energy from the reaction stored in the myosin head. The head then moves into a cocked position and reattaches to the actin. Upon reattaching, the myosin head snaps forward in what is known as a power stroke, pulling the actin and myosin filaments across each other. Afterwards, the myosin head will again detach from the actin, and a new ATP molecule will bind, and the process will begin again. As the myosin and actin filaments slide over one another, they pull on tensile structures within the cell, causing the cell to contract. This occurs simultaneously throughout the muscle tissue, allowing it to contract uniformly. This uniform contraction is vasoconstriction. The second stage of hemostasis is the formation of a platelet plug. Platelets are essentially small red blood cells specifically designed to clot blood when a vessel is damaged, and it involves three major stages. The first is plate adhesion, the second plate release reaction, and the third plate aggregation. During plate adhesion, the platelets gather around the site of the injury and begin to bond via VWF, which is Van Willebrand factors and collagen. Once bonded, the platelets are activated, allowing them to change into the dendritic form, expanding surface area. Platelets carry two granules in them that are released into the bloodstream once active. The first is called alpha granule, and it releases fibrinogen and VWF. The second is called dense granule, releases serotonin, which further constricts muscle cells, ADP, adenosine diphosphate, which activates platelets and promotes aggregation, and calcium, which is needed later for stabilizing the plug. At this point, the platelet releases thromboxane A2, which enables the platelets to interlock together, causing the platelet activation to speed up and activate the coagulation cascade. The third stage of hemostasis is the formation of the fibrin clot, a stronger and more durable seal to the injury. This step can be separated into two main parts, the intrinsic and the extrinsic pathways. The intrinsic is critical in maintaining the formation of the mesh, and the process consists of numerous conversions of enzymes that eventually link up with the extrinsic pathway, aka the clotting cascade. It is important to note that the thrombin speeds out the process of factors 7, 11A, and 5A, subsequently amplifying the cascade so that the fibrin can be produced faster. As aforementioned, working alongside the intrinsic pathway is the extrinsic one, which is more responsible for initiating the fibrin clotting process in the first place. The process starts when the damaged tissue releases proteins that then combine with factor 7A, which converts into factor 10A, where it then converges with the intrinsic pathway. The two cascades come together both to convert prothrombin into thrombin, the very presence of which produces even more prothrombin, meaning it feeds off itself to speed up the production process. This thrombin then converts the soluble fibrogen into insoluble fibrin, which forms a mesh over the injury to reduce further blood loss. It is important to note that there are other factors that are essential to the cascade, such as the presence of vitamin K and calcium. The importance of hemostasis cannot be underestimated, as it plays a key role in homeostasis and injury management. Without it, a simple laceration has the ability to result in hypovolemic shock and ultimately death.